Good evening, everyone. My name is Fahim Shakur, and welcome to this COVID special webinar. It's just gone past 7 p.m. here in the UK, Wednesday, 16th of December. Thank you all for your time, attention, energy, for actually gracing us with your presence. Tonight, we have two world-class research scientists and doctors who are with us, Dr. Mohammed Munir and Dr. Sarah Zaidi from the United States. My co-host, Dr. Mehran Javid, will introduce them fully in a few minutes' time. I just want to introduce the host of tonight. It's MEDtalkx. If you've not come across MEDtalkx, we're available on iTunes. We have a podcast as well as on YouTube as well. And we've been doing throughout the pandemic a weekly show, which has also gone on Pendle 103.1 FM. And that's been on Tuesday nights. And we've been covering a whole host of medical topics that have been free for the entire public, usually about 1 hour in length. In addition to this, we host a actual book club every month, which is also a good spin-off that we've been doing, where we discuss medical and non-medical books. And if you wish to get involved or participate in any of our activities, then please do not hesitate to get in touch with us. We'll leave our contact details in the chat box. Just a couple of things to do with housekeeping. If you're not speaking as one of the presenters or allowed to actually speak, then please could you keep yourself muted and your audio off. It helps with our bandwidth and also to prevent any commotion and distraction to the speakers. We have a lot to get through. We have two presentations, roughly 15 to 20 minutes each, along with the questions as well. Please use the chat function, the chat box, if you've got any questions for the actual speakers tonight. It should be illuminating talk about the COVID-19 vaccines. I've also put the feedback form. Feedback is a gift, as we all know. If you could actually fill that in, it only takes 60 seconds at the end of tonight, and then we can actually help to do better things. And also, Mehran has kindly put the actual web link for the MEDtalkx channel on YouTube as well. So sit back, relax, enjoy the evening. I hope it's informative, enjoyable. You get something out of it. And over to you, Mehran, to introduce our speakers and start the evening. Thank you so much.
Thank you, Fahim, and welcome everyone for this webinar on COVID-19 vaccination. So it's a very important topic, and I'm really delighted to have our two experts. So first to present, we've got Mohammed Munir, who's a lecturer in molecular virology in Lancaster in the United Kingdom. He has research experience on virus pathobiology, viral antagonism of immune responses, as well as having a very keen interest on mechanisms of interspecies pathogenesis of viruses, especially influenza viruses and coronavirus. He has been exceedingly in demand, especially over the past month or two, about his expertise in the matter. So I'm really looking forward to what he has to say about COVID-19 vaccinations, especially a bit more about viruses. So over to you, Mohammed.
Well, thank you very much, Mehran and Fahim, for very kind invitation. This is the first time I'm here, truly intrigued. What I will be doing in 10 to 15 minutes from now on is just to give you a flavor of what are really the trust deficits and what are the reality, what we have as factual responses. So it wouldn't take too long. But before I go into that, I would like to give you a bit of flavor what kind of research I do. So just to get you a bit of idea about kind of work I do in the laboratory. So just to put into the perspective of COVID-19 research, which we'll be talking in a minute in greater detail, we have developed at the Lancaster University a COVID-19 vaccine, which is one of the very few intranasal vaccines, which means it can be administered to people who have hysteria for the injections, or it can be administered very safely to young and elderly people without having any invasive approach. We have completed already its preclinical studies, and WHO has backed up this approach. Hopefully we will be getting into phase 1 and phase 2 trial in February in Egypt, and hopefully the British government would also be on board. And I also sit onto the expert panel on WHO for animal models and diseases, as well as the vaccine. So basically every vaccine that come into the public domain, we go through all of the data and we really make sure that this is what really is in need. Beside this one, I'm very pleased to share that we started working onto one of the artificial intelligence guided diagnostic platform, which last month it got MHRA approval. which is certainly a great step moving forward to roll out into the market. And we have now a private company called Vidya that is not only marketing in the UK, but also around the world. So that's really something that we are very proud of. Beside this one, I just want to thank everybody in my group, very small group, 10 people in there. But the beauty is that we are representing 7 nationalities. That is what where the UK stands for. And we are very proud to present what we work. What we do normally is all about viruses. We don't do anything other than the viruses. And I started covering coronavirus for 500 plus time on TV and radio. Basically from the day when its name was not even identified, it was called mysterious disease. So really just to give you a flavor that I do have some experience, and based on that experience, I will be speaking tonight. The agenda is very simple. 2 questions. Why? First question is why we need COVID-19 vaccine. And then if we really need vaccine, why is there a hesitancy? Is there any factual responses for that? And can we mitigate those myths, and can we really debunk them? So I'll be taking 5 or 10 minutes on each of these to really take you through. The first important point, I think I would like to emphasize onto the fact that if you look onto the disease impact in every year in the last century, it's been enormous. For example, in measles, 500,000 cases were reported, and same is the case with the pertussis, mumps, rubella, smallpox, diphtheria, polio. In that century, just 20 years before, if you ask anybody, somebody would know some of their relatives, those are affected with polio. But that is not really the case any longer. I mean, we have reduced the number of those diseases down to 0 in majority of those cases, and at least 90% plus reduction in those infections. And why that has happened is purely because of the vaccine regime. And that's how we can really see the impact of these vaccines. Another thing that I would like to highlight is that, I mean, probably flu vaccine is something that we all are very aware of, because every year it's been rolled out. Certainly, we didn't have a great level of success with the flu vaccine because of its segmented genome. One virus has 8 segments, another virus another 8 segments, and when they mix together, they can make a new virus with another 8 segments. For whole of my academic career, I've been working on to flu right from my PhD until now, and it's really a challenging virus. And we have had different level of successes each year for the vaccine itself, from 19 to 60%. So on an average, it's a 50 to 60% success rate with the flu vaccine. But still, millions of people are being immunized, particularly at this time when we are anticipating the number of cases start to come due to the winter pressure. But in contrast to the flu jab, which is 50 to 60% effective, we have a vaccine that is 90 to 95% effective. So I don't really see the reason why not to take that opportunity and really to put a good handle on to this pandemic. I mean, the other thing is that, of course, the impact of the COVID-19 itself onto the global population, into the human. You can make comparison with anything that you want to make, but I just wanted to put things into perspective that in 1 year, we have lost 63,000 people in the UK only. And if we compare with the HIV and AIDS causing or AIDS related deaths, the death caused by COVID-19 in 1 year is more than last 20 years caused by HIV combined. So that is the scale we are referring to. And certainly without vaccine, this cannot be mitigated. One second last point I would like to highlight here is that some people said, okay, well, if one person isn't vaccinated, what would the impact it would have onto the society? Before we go into the vaccine, I think I'm going to put one scenario that is a real life scenario, that there were 55 people attending a wedding and only one guest was infected, right? And that one guest, when get back home within the family, within the zone of societal circle, it spread the infection to another 177 people. And that led to death of 7 people out of those 177 who were infected. None of these 7 people were the one who have attended the original wedding ceremony. So basically, what is the fault of these? You person yourself might be healthy, all fine. You might not be getting severe consequences of this disease, but you're going to pass on to somebody who would have underlying condition, your parents, grandparents, and so on. So certainly, if we have the vaccine, we are going to mitigate this problem. One thing that probably people don't really embrace is that the impact of COVID-19 on the livelihood. I mean, the figure onto the left side, you will see the impact of COVID-19 on unemployment in the United States. I mean, it's one of the worst hit countries. And just look onto the figures, onto leisure and hospitality is only 40% unemployment rate. It's massive. And this impact has been relatively little here in the UK because of the furlough scheme offered by the government. So it doesn't really reflect the actual unemployment status. But just to put into the perspective, there has been a very reliable survey that has been conducted to ask the people that if they lose the job because of COVID-19, for how long that can they survive? 35% out of 100 been asked, they said they can only survive less than a month. So that is the impact on the livelihood of any infection like that. So if there is a vaccine availability, certainly that is something to count on and to jump on. And the same is the case with different months. And I mean, only 23% people said that they can live up to 7 or more years. So that is really something that we have to keep in mind. Probably the last thing that I want to highlight here is that we, so far, in 1 year, since the pandemic has started, we have acquired only 20% of the herd immunity globally on an average if we look on. And at the same time, we have lost 1.6 million people. This means that if we carry on with this momentum to reach 60% of the herd immunity required to break the transmission chain, we need another 3 years. And that means 4.6 million people have to die before we achieve 80% herd immunity, 60% herd immunity. And after 3 years, we don't really know if the first person who have got the herd immunity is protected yet or not. So this means the natural course of infection is not really something that we should count on to achieve this herd immunity. The alternative is the vaccine. So here is a graph that here we are standing at this moment, end of December, maybe early January, 20% of the immunity. So if we start rolling in the vaccine that is shown here in the light blue color, that means the vaccine would have impact onto the number of infections. So number of infection would be put a little bit on the bay, and then we will be establishing the herd immunity by, say, end of next year, reaching closer to 60%. And if we reach to the 60%, that is the point when we start seeing the benefit of the COVID-19 vaccine. Until we don't really reach to that point, I don't think that we should be jumping too much. It would be a little too early. Dr. Zaidi will be giving you a little bit more introduction about the mRNA-based vaccine, but I just wanted to make a few points here. The mRNA-based vaccine that is approved by the Pfizer and BioNTech vaccine, that's given in 2 doses, each with 3 weeks apart approximately, and it takes another 1 week before the full immunity is mounted. But that immunity can only protect against the severe form of the infection or the small side of, not the side effect, I mean the small, the lighter version of the disease itself. But it didn't really protect anybody as it stands at the moment from the infection or transmitting the infection. So that also means that even if the people are vaccinated until the herd immunity is not achieved, they still have to self-isolate if they come across with a contact and tracing system, or they have to quarantine still until we have accommodated solid data that can demonstrate that the transmission can be reduced a little bit. AstraZeneca vaccine is probably the best shot to have a bit impact onto the transmission, but that is something that we have to watch for a while. How long the immunity lasts, we don't really know. We certainly have a data for 2 to 3 months immunity, but based on our previous experience with coronaviruses, it's not something that we should count more than 2 years the most. So with that said, I just want to take you to 8 of the points that I have accommodated, not only just being asked over the media, but also people who have a concern about the vaccine hesitancy. And I'm going to take you through a few of these points in relatively more detail to just give you some sort of like relevance to the vaccines, particularly speaking about the Pfizer and Moderna AstraZeneca vaccine, and whether those claims are true or false, and to really give the factual responses against that. Probably the first and the foremost is that people say that any of the vaccine that is introduced into our veins is going to be tracking us, collecting biometra, or maybe really getting the information that we don't really want to do that. I just want to simply state that that is false. That is not really true. And that certainly has relevance toward the Pfizer and Moderna, because AstraZeneca vaccine is one of those that is a DNA-based vaccine. Why it is not relevant? For one reason. I think the audience on this platform, they are fully aware that our whole DNA is impregnated, is very much condensed within the nucleus. In the cytoplasm, there isn't any DNA. If the DNA is in the cytoplasm, there is a pathology. So this means that anything that go into the DNA might have some impact. Okay, anything that remain into the cytoplasm, they cannot be integrated into our genome. Looking onto very specifically onto the mRNA-based vaccine, these are just a small RNA that is encoding the spike protein encapsulated within the nanoparticles given to the cell, so that the nanoparticle delivered to the cellular translation machinery. And when the protein is expressed, that is presented to our antigen presenting cells. And those antigen presenting cells then start taming the T cell, and then T cell make the B cells, and B cells start producing antibodies. So the whole objective of the vaccine regime is really to produce antibodies as well as the T cell responses, right? So if we really look onto this vaccine, the Pfizer and BioNTech vaccine is mRNA-based vaccine, and mRNA that is, when it is delivered into the cytoplasm here, and the one that is shown in the orange red is very short-lived, because RNA itself is a very fragile molecule. It doesn't stay more than 4 to 5 days. So it will be cleared off by our regular clearing mechanisms such as apoptosis or so. And the mRNA doesn't go into the nucleus anyways, because it doesn't need to. It needs to be translated into protein, and that only can happen into the cytoplasm. One of the important, which is a little more technical, but I'm sure you will be able to understand, because I can see that a lot of people are with the scientific background here, is that the RNA is a different molecule than the DNA. RNA is a ribonucleic acid, and DNA is a deoxyribonucleic acid. So they are not compatible. RNA has to first convert into DNA before it is a possibility to get into the DNA integration. And this conversion of RNA to DNA is possible because HIV or other retroviruses can do convert, but that person has to be infected with that, and it has to have that element that can convert RNA into DNA. So all in all, this is extremely less likely. It's not going to happen. One of the things that probably very few people know how the Oxford AstraZeneca vaccine work. It is a little more complicated than the mRNA-based vaccine. It is based on a vector vaccine, which is a chimpanzee adenovirus, normally cause common cold in chimpanzees, but this is mutated in such a way that it does not express some of the essential proteins. And because of this inability to express those proteins, it does not replicate, it just stay as it is. So the spike protein of the SARS-2 has been cloned into one of the expression vector that is driven by the promoter acquired from the CMV, which is cytomegalovirus, and it has a TET repressor. So what TET repressor does is that it just keep the whole mRNA for the spike protein under the bay. It doesn't let them express until you add the oxytetracycline into the cell culture. So once you add oxytetracycline, TET repressor go away, the mRNA for the spike protein start to produce. And then once the mRNA is produced, it's the same as mRNA-based vaccine. So this replication deficient chimpanzee adenovirus is very ectopic. It only come into the cytoplasm, and it doesn't go into the nucleus. Since it doesn't go into the nucleus and it doesn't replicate itself from one cell to another cell, it cannot be integrated into the genome. So this is completely false and should be completely ignored. One of the things is that people are asking about the vaccines are made into some sort of lungs or the cell that are taken from the aborted fetus. For the scientific community, it is pretty clear that the vaccine vectors have to have some media to grow. Viruses behave very differently than the bacteria. Bacteria doesn't require any medium to grow. They can grow on their own, but the viruses, they have to have the cells, and we have to acquire those cells from one source or another source. Mainly embryonic or stem cells, they are more permissive to the viruses. Therefore, we get a good quantity of that. So that is certainly true. And it is applicable for the AstraZeneca vaccine because that is produced into HEC293 T cells. But when we talk about the mRNA-based Pfizer, BioNTech, or Moderna vaccine, that is purely synthetic. There is no any cell culture involved, so it is completely acceptable for people who are having concerns about with the aborted fetus. But the important thing that I would like to highlight here is that AstraZeneca vaccine or vector-based vaccine, they required cells to make more vaccine of their own. But they use some of the cells that are originated from the aborted fetus. But the first thing is that those fetuses were not aborted just because of the vaccine production. And the second thing is that when the virus are grown into the cell culture, is purified, and only the purified virus particles are given as a vaccine. There isn't any cell going along within the vaccine. Just three next point very quickly is that some of the objectionable substances in the vaccine. Certainly, like for example, flu vaccine has gelatin because it is required to stabilize the virus. And there are some concerns that some of the other vaccine for the COVID-19 could have some of those objectionable substances. But as it stands, when we talk specifically for these three vaccines, there isn't such thing that we need to worry about. Vaccine or genetically modified substances, of course, because we have to modify them to express the foreign gene, and that's how it all works. But genetic modification isn't really new for us, I think, in this era. Everything we eat from banana to strawberry, everything is having one or other way of genetic modification. The mRNA-based vaccine is certainly suitable for vegetarian, and it's called halal vaccine is because it's purely synthetic. There is no animal or a human source involved within that. One of the things that probably I've been asked the most is that at one end, Pfizer, Moderna, and AstraZeneca, they keep asking that the vaccine is safe, step up, get the vaccine, but at the same time, they do apply the indemnity to the government. So why is that case? Are they worried or concerned? Well, not really. Part of the problem is that the vaccine industry itself hasn't been very profitable, except few of the companies who managed to get the vaccine approved in the early stages for influenza or so. And so there is 90% chances that the vaccine that are being developed might not work. So therefore, there is a heavy risk involved within that. So because of this risk, pharmaceutical were not jumping into getting into the business. So of course, the government have to ask them to come and do the business. They have to have some sort of assurance. And any disability caused by the vaccine has to be covered by the Vaccine Damages Act. In the UK, it is 1979. That means if any damage occurs partially or fully, has to be compensated by £120,000 one-off money. And that's an enormous amount. I mean, if you look onto the history of 40-year, UK government has paid £80 million in this compensation. And that is purely to attract the pharmaceutical to come into the country and really get the business done. So it's not because of the vaccine concerns.
Mohammed, can I just give you 2 minutes to wrap up, please?
Yes, absolutely. I'm getting there. So one of the reasons that is being developed very fast, I think in the scientific community, we do know that is not really the case because we have a history of 17 years of knowledge working with the coronaviruses, and we have exploited that definitely. Some say that mRNA-based vaccine is really new, but not really the case. I mean, if you look onto this graph, you can see that the first mRNA-based vaccine went into the human arm in 2013. So we have at least 7 years of history of the safety. Just the last slide I would like to put up over here is just finding the full balance between whatever I said so far. On one end, what is my daily risk? The risk is that 3 people are dying every minute, given the number of infection and the mortalities we are having so far. And on the availability side, we have a vaccine that is 95% effective, and that is tested on more than 30,000 people, and all those are doing good, except 2 cases, those were not really associated with the vaccine itself. So with this finding sort of balance, I always say that the choice is yours, but it's really good shot to take. Thank you.
Thank you so much, Dr. Munir. That was a great presentation. As Mehran said, we're just going to take a couple of minutes for some questions and then move on to Dr. Zaidi's presentation. Dr. Munir, if I can ask you to stop sharing your screen so Dr. Zaidi can get her presentation ready as well. And in that switch or time for transfer, you can take a few questions as well. So we've got a few questions, and I think one was to do with effectiveness. One of the participants said, what does this 29% effectiveness mean in regards to the flu vaccine? And I think they went on in terms of prediction for 60% herd immunity of vaccine assumes 90% effectiveness, but it's already mutating. How likely this prediction will be realized? I think you need to split that into the two components, Dr. Munir. There's a degree about talking about this mutation, which we heard about in the news the last day or two, and if you can actually explain that to the audience as well.
Yes, absolutely. Well, vaccine is something, the bottom line is that you don't need to worry about, because since the virus has emerged, we have more than 12,000 mutations in this virus. And none of them has any significant impact onto the disease severity, transmission, or the vaccine effectiveness. When the UK government announced that there is a mutation going on into the virus in the UK, they didn't really precisely tell what is happening. But since I work into the UK Genome Consortium, so we explored all 50,000 sequences, those were sequenced from the UK. We identified only one mutation that probably have a concern, that is N2Y mutation at 501 position within the spike protein. That is not in the receptor binding domain, that is not in one of the T or B cell epitopes. So therefore, this mutation wouldn't really have impact. I have a strong feeling that there was other political reasons that this has been put up, and I think yesterday I discussed this one on BBC, that there is no reason to believe that this mutation would have any impact onto anything that we can think of.
Great, thank you, Mohammed. Next question, and we've had lots of questions coming in, but I'll try to get your opinion about what is the difference between natural immunity as well as immunity that's propagated by having a vaccine, and with special focus on primary infection as well as secondary infection, because there are some cases where you can be infected twice.
Yeah, I mean, we know with the history of the vaccine is that the natural course of infection of any virus doesn't induce as much immunity as is induced by the vaccine. And that is primarily because the vaccine is given usually in 2 doses, in prime and the boost, and the expression of the antigen is in large quantity. Therefore, our immune system is mounted relatively strongly. So therefore, the current recommendation is that if people are infected with COVID-19 and they recovered, they still have the antibody, they need to be vaccinated, because the natural infection acquired immunity might not sustain longer, because we are seeing the reinfection cases.
Excellent. There's so many questions, but time is always against us. So we're going to take a couple more, then go on to Dr. Zaidi's presentation. Then we'll have the floor for you both to mop up any further questions. All right, Dr. Munir. So the next one was to do with, did I hear correctly that the vaccine only gives immunity for 3 months? So I think this question is quite interesting in that actually, how long is the immunity for, and do people need a top up and so on? Can you actually combine that and answer that question in one go?
Oh, you're asking me?
Yeah, for you.
Yeah, no, what I meant was that we have the proof that the vaccine induced immunity would be at least for 3 months, because that is where the phase 3 trial started and until it is concluded. So that is for sure we have it, but it is going to be certainly staying more than 1 year based on our experience with the SARS and MERS. So the people who were naturally infected with the SARS, they have the antibodies more than 1.5 years, but we didn't really know that it was protective enough, because at that point the virus was gone. So I have an anticipation that if it is not going to be 1.5 years, at least it's going to be for a year.
Great. And just one last question before I introduce Dr. Zaidi, who's patiently waiting. Just going back to one of the questions about the word effectiveness. What does that actually mean? And how is that different to, say, just being effective?
Yeah, basically, in the scientific term, there are two. One is the efficacy, another is effectiveness. When we talk about efficacy is something that you determine under the controlled environment. For example, the phase 3 trial was basically a controlled environment where people are self-isolating, people are having masks and covering. So efficacy would be different than effectiveness. Effectiveness is that when you take the vaccine, bring into the field, and in the field, whatever the coverage comes, that would be effectiveness. And we know that based on our previous experiences, whatever the vaccine efficacy you get in phase 3 trial, that would be 10% less into the vaccine effectiveness. So basically 95% would turn out to be around 80-85% in the field. That is what is effectiveness. And that is really the big thing that we have to count on before we really saying that, oh yeah, we got a vaccine that is 95% effective.
Fantastic. Thank you, Mohammed. So I'd like to take this opportunity to introduce our next presenter. So this is Dr. Sarah Zaidi, who's a public health specialist, and she's also the co-founder of the Center for Economic and Social Rights, and served as a science and managing director for about 13 years. So she is a double Ivy League graduate. I guess my relationship with her is because is through being the co-editor of the Breaking Down COVID-19 textbook, which is free and available for people. So we will try and put the link on the chat box for you to have a look. And there's a great deal of knowledge. I've certainly learned a lot through that experience. So over to you, Dr. Zaidi, for introducing the vaccines update.
Thank you. Thank you, Fahim. Thank you, Mehran, for inviting me. And Munir, that was a brilliant presentation. I'm nowhere near an expert like yourself. I started working on COVID-19 earlier, just before, around the outbreak, basically, in December when it was first emerging, and just generally following it. And we then started working on this book, which is Breaking Down COVID-19. I'd encourage everyone to go and look at it. It has 24 chapters. It's pretty thorough. And our intention is to constantly update it. But coming back to the vaccines, now let me see how to work this. So my talk is basically around 10 points. And a lot of this has actually been covered by Munir, so I'm not going to spend too much time around it. But it's been very promising, this vaccine development timeline, because if you recall, the first case of COVID, the cluster of cases just emerged about a year ago, right around this time. And it's really encouraging to see that now the vaccines are actually being injected into people, and it's one of the fastest development we've ever seen. Why is that? Well, we actually have quite a bit of history with coronaviruses, and there's been investment in developing vaccines for these viruses. But of course, since we never had enough cases, we were never able to test them. So that's one reason why there's been this huge history in terms of knowing and warp speed development around the vaccine. This is a novel pathogen, and therefore the amount of damage it's caused because of its transmissibility has been quite huge, especially with the lockdowns and bringing economies and travel and life to a complete halt as we've known it in the 21st century. And it's a very interesting virus because it also disproportionately hurts those that are older and causes asymptomatic or milder diseases in younger people. And therefore, sometimes people don't see the benefit of having to use public health interventions because they're like, we're not being heard by it. But it has a very high mortality risk for those that are older and those that have chronic disease. It's actually disproportionately affected disadvantaged communities, Blacks, Latinos, and other minorities. And why is that? Because this virus is easily transmitted through airborne particles and by people who don't appear sick because they either don't develop the full sickness or they're pre-symptomatic. And therefore, it's very important that if we are to defeat this virus, given that we can bring it under control through just public health measures or what we call non-pharmaceutical interventions, vaccines are very necessary for us to control and defeat this pandemic. So how do vaccines really, what are their benefits? They prevent the infection because they imitate the infection inside our body and they trigger the immune response. As you saw Dr. Munir point out, they're very helpful, and other diseases that we've experienced have really been eliminated because of that. They also help to prevent severe disease and death after the infection. And eventually you want them to reduce the transmission and just prevent it altogether so that the disease is gone, like in the case of smallpox where we vaccinated everyone and the disease is no longer around. So for the COVID-19 vaccine, normally you have 4 phases to these vaccines. You have a preclinical phase where you look at and determine the safety of them in animal models. Currently, we have 86 vaccines under there that are being looked at. You have the phase 1, which is the smaller trials for people. There are very few dozen people. 41 vaccines are being investigated. Then we have the phase 2 and phase 3. So we have hundreds, and then in phase 3, 1,000. And then there's the phase 4, which is the ongoing safety and assessments for multiple years. Now, coronavirus vaccine basically looked at trying to prevent severe disease and prevent hospitalizations. I mean, that was the purpose. There were so many people that are being affected by it that the designers were interested in looking at death and looking at prevention of hospital admissions at the end. So there are 13 vaccines that are very promising, that have made it to the phase 3, and actually many of them already are currently under limited use. So here we are with other vaccines, and this coronavirus vaccine, why it was so rapid, I think because also many phases were actually done very in parallel rather than sequentially. So, while the vaccines were being looked at, many of the phases were combined. 1 and 2 were combined, 2 and 3 were combined. And that's been a very, has led to, and there was also a lot of investment in these vaccines. The Operation Warp Speed alone is close to $11 billion. Then you have CEPI, and you have other governments who've invested quite a bit in vaccine development. Here's the sort of a quick timeline from the first set of clusters that emerged on December 15, 2019, to the time that we actually got the genetic structure of this coronavirus, which actually speeded up not only vaccine development, but also the diagnostics. To the first set of clinical trials began in humans in March. March 16th was the first sort of Moderna had started testing their phase 1 clinical trials. July 27th was the large trials for Pfizer and Moderna. And then, vaccines were, first set of the results came in the middle of November. And within a month, you had the approval, and soon after we had the first person to be immunized. But meanwhile, there are other vaccines, mostly inactivated or the adenoviral vector vaccines that have been approved for limited use in China, Russia, and United Arab Emirates. So how are vaccines designed? And this is a very simple way that I'm going to show you. It's a lot more complicated, but this is very quickly. So what we're looking at is when you have a new pathogen, there are no antibodies. And with coronavirus, there was no antibody, and therefore we needed to look at what the antibody, which is the antigen that is the active one that was binding onto the cell and infecting the cells. Very quickly, we learned that was the spike protein that was looking in. Most of the vaccines are looking at least at the portion of the spike protein. Then we looked at the various platforms. Now we have 6 or 7 platforms, basically. We have the inactivated, these are the classic approaches that we have been using, and many of the Chinese vaccines are based on that. And then you have the viral vector vaccines, which are sort of a modified platform. That's the Oxford AstraZeneca, the CanSino, and Sputnik 5, which are using various adenoviruses. And then you have the more modern messenger RNA platforms, which right now we have the Pfizer, BioNTech, and Moderna vaccines. And I'm not going to spend any time because I think Dr. Munir explained this very well of what happens with the mRNA vaccine and its safety. Basically, it's it's the lipid in which it's encased. That's the big difference between the Moderna vaccine and the Pfizer vaccine, because we don't know what the lipid is, but that's how they enter the cell to make it more stable. Then these vaccines, all the 13 vaccines that are currently in phase 3 clinical trial, are also all injectables. And the doses are repeated to be more effective. And you'll see in the next, not this one, the following slide, I'll show you why you need 2 doses. But basically, most of these 3 vaccines are 2 doses, 3 to 4 weeks apart. And the clinical trials have been run on very large number of people in various different sites, which is very promising. So in most of these vaccines, in fact, in all of them, other than the Oxford AstraZeneca vaccine, we've seen 95% efficacy, and we've seen almost in the Moderna vaccine, we saw 0 severe cases. In terms of safety, they've been, mild symptoms, usually within the first 7 days of vaccination, nothing serious. The issue really is about storage. The Pfizer vaccine has to have been in very cold storage. You can only stay in the refrigerator for 5 days and has to be used within 6 after being diluted. Moderna is slightly better. It can stay for 12 hours at room temperature. So it's really, in terms of delivery, becomes of a challenge. The Oxford AstraZeneca vaccine can stay at 2 to 8 degrees centigrade. We have many doses that are coming, which is great. So we can technically start to do the high risk communities, populations can start getting their vaccines. So here is in the FDA, they shared this graph, which showed that in the vaccinated group, within, even after the first dose, if you look at it, the blue dots are the number of cases of COVID infections, whereas there are so few cases, incidences of COVID infection after in the Pfizer vaccine. Side effects, nothing really serious. People, I think the most serious was Bell's palsy in 4 cases where there were 0 in the case of the placebo, but apparently not related and resolved. The swollen lymph nodes also were resolved. Really nothing, there were 2 deaths also, but they were not related to the vaccination. Now, these vaccines did not really test for transmission, and it's not known whether a vaccinated person can still be an asymptomatic carrier of passing on COVID to others. And the only one was the Oxford AstraZeneca that said they could reduce transmission, but as more data comes in, we can easily measure infectious rates in the vaccinated group versus the placebo group to see whether they will be effective. There's no reason to think they won't be effective, but thus far, they're only looking at severe symptoms and prevention of illness. I won't spend any time on herd immunity because Dr. Munir covered it, but you need to have a relatively high number of people vaccinated in order to get community level protection. And I think that's it, but this is very promising. So if you look at it, there are so many vaccines coming with very different platforms. I think Dr. Munir mentioned the intranasal vaccine, which are also being investigated. So I think hopefully by September or earlier, we might have this pandemic under control. Thank you.
Thank you so much, Dr. Zaidi. That was a fascinating presentation. I learned a lot. Something you alluded to, which I will reiterate, is all of the information that our presenters have given is accurate as of today, December 16th. Things are obviously moving with days, and we're learning more and more about things. So today is December the 16th, and it's important to keep that in mind. What I'd like to do is obviously add Dr. Munir to the discussion if he can also put his video back on and unmute himself. And then we'll open the floor to questions both for you, Dr. Zaidi, and for Dr. Munir. I appreciate that we're running out of time, but we will go on a bit extra just to cover as much as we can, but we will definitely end within 25 minutes. So first question for you, Dr. Zaidi, in no particular order. The first one is really this time frame for a vaccine to be proven successful. What is this general time frame? I think there's something to untangle there, because people have noticed vaccines have taken longer in the past, and this one's a lot shorter. So can you tell the audience a little bit about the time frame needed for a vaccine to be proven successful?
Well, I mean, the normal time frame, as you saw, was about 10.7 years. I think with this, because of the investment, because it was there's huge amounts of monetary investment, and I think we also had a leg up in terms of having studied other viruses. You don't forget, there's been Zika, there's been MERS, there's been Nipah virus. There have been vaccines that have been developed. The problem is those infections have disappeared or have come to a level that we cannot run those clinical trials. And in this case, most countries could not control the infection. So it became, there are so many cases that was easier for these pharmaceutical companies to run their trials on a large number. And I think that's made a huge difference.
Great, thank you. Thank you, Sarah. And another question, just with reference to your slide about the side effects between the different age groups that it kind of defines. It is quite interesting that those age 55 and above appear to be on average either similar or less in terms of their prevalence of side effects. Do we suspect that the numbers are underpowered?
I'm not quite sure. From what I understand, and maybe Dr. Munir can speak to it more, it's also that the immune system of elderly is quite different than the immune system of young people. And because this is a controlled amount of virus that's being introduced, it's one reason why there might be fewer side or lesser impact of side effects. But Dr. Munir, maybe you have a better response.
That's right. That's right.
Good, excellent. We like short answers as well as long. Dr. Zaidi, further question. And it's a bit of a contentious question, but I think you should actually try and address it on the head. Why are people suspicious about the current COVID vaccine? I know Dr. Munir talked about vaccine hesitancy, but that's slightly different to suspicion, mistrust, distrust. Is there anything you would add to what Dr. Munir said about vaccine hesitancy on this point?
Well, from being based in the US, the whole experiments that have often been done on minority groups makes people distrust vaccine development that's been so rapid. And even the way I'm from Pakistan, if you look at the way polio was used to get to Osama bin Laden, people really there's a whole history, political history for why people distrust vaccines, which is rather unfortunate. We also have the whole link with autism that was, about 15 years ago, where certain vaccines, I think it was, can't remember, it was Hep B or someone that had, there was suspected links that didn't really pan out after a while. So I think there is a reason for why people often distrust vaccines. Which is rather unfortunate because they are very effective. It's a very controlled amount of, pathogen that's introduced into a person. It's so much safer than getting natural immunity. You're getting naturally infected by the virus where you don't know how the trajectory of this virus is going to play out.
Great, thank you, Dr. Zaidi. Mohammed, if I come to you more about recent events around the new strain that they suspect may have emerged in the south of London, south of England, I should say, and around the London borough. What do we understand by that? And are we expecting it to continue to evolve over time?
Yes, absolutely. We should not underestimate that the viruses do mutate, and primarily when they mutate, they mutate for their own benefits. But the important thing to highlight here is that when the first when SARS-2 started infecting us back in December, at that point, we didn't have any immunity against the virus. So we were completely naive. So virus didn't have any resistance against. So as we moved along, we have like 15 to 20% people are already immune to in one way or other. Then the virus has a resistance. So it is a immunological pressure on. So virus has to mutate, and it will mutate quite frequently. And then what will happen is that, I mean, it's already happening in many of the countries that few of the strain become predominant strain. So those become reference strain for each of the countries. But those mutations that we see are literally having no or little impact. The only mutation that can have impact is on the spike protein. And then within the spike protein, there are certain mutations. If those are onto the epitopes or those on the receptor bindings domains, those are the ones that need to be looked for the transmission severity of the disease or for the vaccine effectiveness. But beside that, everything else is all fine. Stay cool and calm.
It's good advice for a pandemic. I like that. Dr. Zaidi, how are vaccines tested before given to humans? I know that's a broad question, but I think what that's getting at is that the questioner would like some reassurance that we're not guinea pigs getting this as such. And
No.
So if you can just tell us a little bit about the process before it's actually given for trials.
Let me say, I think Dr. Munir will be a better person to answer that, but they're basically on animals and he'll be able to tell us more about it.
Yeah, sure. I mean, basically, we have just finished that study for our vaccine. So it's called preclinical study. It has to be conducted on animals, and in the WHO, when we work on, we define which are the best animal models to study the vaccine before it go into the human arms. For SARS-2, there are two options. Either you have to do in non-human primates like rhesus macaque, or you have to do in any other two animals like mice or in hamster. So you got to choose which model you want to go for. So it has to go through the animal route, and all the vaccines that are in the front line and advanced development, they all went through the same pipeline, because until those are not tested preclinically, they can't go into the human trial. So that is the process. Rest assured that all the vaccines that go into the human arm, they are rigorously tested and pass through all the stages that we otherwise do normally. And I want to make a point here is that the vaccines that we've been developing before the SARS-2, those been tested on less number of people compared to what that has been tested here in the SARS-2. So, I mean, 30,000 people have hardly been included in any of the phase 3 trial alone. And so this is really something that we have to shouldn't take for granted. It's just the unprecedented need that pushed the timeline.
Great, thank you, Dr. Munir. If I come to yourself, and perhaps Dr. Zaidi can chip in, there is a question about choice. How are the general public, or I guess the authorities, educated about which vaccine to use? Because let's say, for example, Bahrain, they've approved both the Chinese vaccine as well as Pfizer, but when you've got 200 vaccinations under study from phase 1 onwards, I think only 13 have got that higher tier of approval. Who are the regulators? And is this just going to be done on a local or national basis?
You want me to answer? Or?
Yeah. Yes, please.
Sure,
yeah.
Sure. I think this is a fortunate event that when we are expecting more than one vaccines coming through, that hasn't been, very common for other diseases. For example, when Ebola came, we know that there have been few front runners, but the choice was very limited. But the beauty here is that we have vaccines; those are of different technologies. So, for example, mRNA-based vaccine is likely suitable for immunocompromised people. I don't see any problem going into that. And then the vectored vaccine or the live attenuated vaccine would be the one, those would be challenging for people who are on a chemotherapy or on a cancer treatment and so on. So therefore, when a country would approve more than one vaccine, it's not, I don't think it should be by choice, it should be by need. So maybe they disperse the safer technology to a certain cohort of the people, and then the rest go on to the other cohort where it has been tested and given weightage more.
Dr. Zaidi, do you want to add anything to that?
No, I mean, I think this is actually quite unprecedented to have so many different platforms and choices of vaccines. I think for an average person who's not immunosuppressed or suffering from any kind of chronic illness, it's take what you can get at this point. I mean, the 30,000 in Moderna, and keep in mind that this data came in November, 44,000 for Pfizer, and we're going to continue to see these numbers hopefully go up and to see longer trajectory of data coming in. But I think the side effects are quite minimal. Even from the CanSino vaccine that's being currently tested, which is adenoviral vector vaccine, it's AD5, we're seeing very promising results in terms of disease severity. So, I think if you're not in a group that needs special attention, I think you can choose and be secure in taking whatever vaccine is available.
I just want to finish off by stating that we need at least 13 billion doses to make 70% herd immunity in the world. And I don't think that is per granted. And the major problem is the low and middle income countries, to be honest. And COVAX, which is a facility set by Gavi and SAPI and World Health Organization, the only thing we can promise them is the 3% of their population. And that would still account for 2 billion doses, which we don't have the capacity so far. So it's not about the money availability and all of that. It's all about the infrastructure. I mean, Pfizer, Moderna, particularly speaking onto the Pfizer, because that is now approved in at least in the UK and a few other countries, they have a maximum capacity of 50 million doses in this year. So even if you pump in $1 billion, it's not going to help them because their capacity is more than that. So what we just going to wrap up is that, as Dr. Zaidi said, whatever is available, take it if this is something that you got to choice.
That's good advice. We've got a couple of people asking a similar question, so I'll come to you, Dr. Zaidi, first, and if needed, Dr. Munir as well. And it's to do with women of childbearing age and pregnant women. I think that's a good subset of the population that needs to be mentioned here. The RCOG, for our worldwide listeners and viewers, that's the Royal College of Obstetricians and Gynecologists. The questioner have said that women who are planning a pregnancy within 3 months should not have the vaccine. So what are the risks for women who are planning a pregnancy or are pregnant, and what is the advice to be given to them, and when can we expect safety data in pregnancy? So all to do with pregnancy or getting pregnant and the vaccine. Dr. Zaidi first.
Well, none of these vaccines have actually been tested amongst pregnant women. I mean, it's for right now, I wouldn't say they're safe at all for pregnant women. They haven't really been tested under 16 in adolescents and young children. We don't have any data for the immunocompromised. So there's a whole subset of people that these vaccines need to really include these groups in order for them to be tested and to look at their efficacy. I mean, really, the given where we were with this pandemic back in March, May, with such huge amounts of deaths. I mean, the whole purpose of these clinical trials in Rush was to really prevent severe COVID and to prevent people from entering the ICU. And so that was really the end point that people were looking at in terms of developing their strategies for these clinical trials. Now that we have that data, I think now we're going to start looking into other populations.
Good. Dr. Munir, do you want to add anything to that?
Yeah, absolutely. I mean, that's absolutely right. There is no risk of any vaccine so far in pregnant women. It's just because those vaccines have not been tested. Therefore, those are not recommended. That is as simple as that. But I just want to say one thing that some people might have an idea, because there was a German physician who came up with the claim to the European Medicine Agency that the Pfizer and Moderna vaccines can cause infertility in a woman. Therefore, they are not recommending that. And he made a claim that there is a syncytin-1 protein that is very similar to the spike protein, one of the region, and the antibodies raised against that would be targeting the spike protein, and the syncytin-1 is an essential component of the placenta, and therefore it's going to be a problem. It's all debunked. That has no reality. So the only reason that it is not recommended in pregnant women and children under 16 is just because those were not tested in.
Great. And just following on the topic of efficacy, I know I've raised this before, but there seems to be still some lack of acknowledgement or understanding about what that actually means. Does that mean there's no symptoms, there's less symptoms, reduced mortality, or reduced ICU kind of admissions? Because I guess the some of the feeling is that that isn't very clear in how that's demonstrated. So if I ask Dr. Munir for that one.
Yeah, so it depends actually which vaccine you refer to. As Dr. Zaidi was alluding that before the trial was set up, there were certain benchmark those were defined that any vaccine that will take this mark, they will be approved. So it's not necessarily what has been approved for Ebola would be approved for the SARS-2. It's just because of the unprecedented need. So for example, for the Pfizer vaccine, the benchmark has been set that if it prevents anybody from severe disease, it would be considered as effective. Okay, and then when the efficacy on the interim data became like, okay, 90 to 95%, then they put next threshold. Is it going to protect against milder version of the disease or not? So when the calculation was done onto the vaccine efficacy, not effectiveness, vaccine efficacy, that was done onto the milder cases as well. So it just evolved over the time. But is it basically preventing somebody getting severe version of the disease or reducing the risk of hospitalization or unfortunate death?
Great. And I know we've talked about the different vaccines. I just wanted to bring you in, Dr. Zaidi. Is in terms of actually the process of delivery, do we expect Oxford vaccine to be the most adaptable and the most feasible?
Yes, definitely. I mean, look, in terms of cold storage and the chain, et cetera, it's definitely going to be most likely. I would think many of the Chinese vaccines will also be more applicable for low and middle income countries. India is also developing some vaccines for local use. So these are all inactivated viral vaccines or the viral vector vaccines, which are, more easy to transport. But as Dr. Munir said, do require, they need to be manufactured, and that's where we might have some logistical issues. The mRNA technology, mRNA is just very unstable. That's the problem. And for the cold chain we don't have the refrigeration or the cold storage, or even, in most places to be able to administer that kind of vaccine in a timely manner. So I think some of the other more promising vaccines that might be coming in 2021 are also, we're still waiting for the Janssen vaccine, which is also a Novavax vaccine, CureVac. I mean, there's many other vaccines that are also being looked at that might be more appropriate for low-income countries.
And Dr. Munir's vaccine as well, of course.
Yes. Yeah, absolutely. Yeah, indeed. I just want to add a little bit more point on Dr. Zaidi's comment is that I think one of the major issues with the COVID-19 vaccine would be the scalability. And the AstraZeneca Oxford vaccine has certainly a lot of edge onto the scalability, because they set up, right from the beginning, they set up a 10 production unit around the world. And they have a capacity to make billions of doses, and much more than Pfizer and Moderna combined. And that was the big component of the COVAX facility for low and middle income countries. So we basically were, in developing countries, were watching for the AstraZeneca. But they come to more of their lack of professionalism. The problem at the moment is that they are in a dilemma. I mean, put actually scientists in the dilemma as well, that they have a 62% efficacy with 2 full doses, which is certainly not very promising and convincing. And they have 90% efficacy in 1/2 and 1 full dose. And that has been tested on only 2,734 people, all under 55 years old. So that data isn't sufficient enough. So they got to do a trial that required another 3 to 6 months. Unfortunately, that is not something we have to heavy count on. But this is something, if I would have a choice, I would go for the AstraZeneca, because it has so many other advantages that have been proven safe.
Oh, okay. I think that's muddied the waters a bit with the levels of effectiveness, but you'd go for that. I need to rewind this tape and watch this again, I think. Okay.
I mean, do remember we were like looking at 50% efficacy as the threshold for when we would approve a vaccine. So it has that threshold. I mean,
yeah, absolutely. It has a threshold, but actually in the WHO, what we calculate that the cost for the vaccine is almost negligible compared to the operational cost. So if you have a vaccine that is 95% effective, even if it is £10 expensive, that is worth rolling out into the market because all the operational cost would be the same. And then you will get the higher efficacy. But with the low efficacy, the problem is that it's going to come into, because, for example, the Sinovac and the Sinopharm in the UAE, Bahrain, all they have like 82% or so efficacy. So even if we take the average of 92 and 60 of the AstraZeneca, still it is lower than that, is 70%. So that is really the major challenge. Of course, I, being in the UK, would love to have the AstraZeneca, but unfortunately, that is the situation.
Great. Dr. Munir, let's carry on with the questions. We had some that came before we started, so I want to take those and be fair to them as well. The question was interesting. It said, in terms of the vaccine, we know that we could both be infected by the virus and still transmit the virus, even though we have had it. The vaccine protects against severe complications of the disease only, in brackets. So the question is, why then should a low-risk individual who is unlikely to get a severe form of the disease but can still transmit it to high-risk people, regardless of whether he has had the vaccine or not, take the vaccine? Can you answer that, Dr. Munir?
Yes, absolutely. I think basically one of my whole slide was particularly for this question is that it's not about you getting infected. It's about getting those people infected that might have severe consequences. And also, some of the vulnerabilities are not really known. I mean, I was allergic as have an allergy to the dust, and that led to congestion in the chest. That took really, like, I think the first time I came to know was about when I was 18 years old, when I came to know I have a dust allergy. So there are vulnerabilities that we are not really fully aware of, and that lead to complication on the top of everything else. And then we don't know what are the really long-term complication with a long COVID that would develop over the time. So all in all, the final point I want to raise here is that when we say that these vaccine doesn't protect for shedding the virus, that actually not really what we mean. What we mean is that the data at the moment doesn't indicate that. But over the time, when more and more data would be collected from, because you can assume that the first doses of the vaccine in the UK have just been started. So I think even the vaccine induced immunity hasn't yet been developed. It take 1 month from the first dose, and afterward we will be in the position to detect whether it could protect from those people for shedding the virus or getting the infected or so on. In the phase 3 trial data, that is also being looked at. So all cumulatively, it would probably end up that yes, it does prevent, maybe not sterile immunity, but that can prevent complete shedding of the virus, but to some extent. So it would have ultimately impact onto the disease transmission, because otherwise scientifically that is not correct.
Mm-hmm. Good. And we also had another question come in before, Dr. Munir. I'd like you to take. There's two questions, really. There have been 2 reports of anaphylactoid type of reactions in healthcare staff. Those of us who are in the UK, we've heard about this with previous allergies. They had EpiPens, I gather, as well. And what excipients or active components could cause this type of reaction? I think it was created a bit of consternation and discussion. So that's something. And also the 2 vaccinations are given 3 weeks apart for the vaccines that need it 3. I noticed, I think some in process actually 1 vaccine, but the ones we're discussing is 2. Do we know if the response is delayed if the second vaccine is given late, and what to do if it's missed for several weeks? So really about the anaphylactoid and what happens if you miss or you delay your second dose.
Yeah, the first question about the allergies, the first thing is that we don't precisely know, and probably I'm not allowed to share all that information, what is really the component and the composition of this Pfizer vaccine that caused this allergic reaction. But what I can tell you is that mRNA itself wouldn't cause any allergy, because our cell has milligrams of RNA being produced on a daily basis, basically. So that is not any different than that. And even the mRNA that is used in the Pfizer vaccine, that is nucleoside modified. That means our immune system, the innate immune system, wouldn't detect it. It would be the adaptive immune system that would detect it. More likely, it is associated with the nanoparticles or the composition of the nanoparticle that is there. What I can tell you is that the nanoparticles are composed of 4 different components of the lipids, and the lipids are part of our own body. Okay, so it has a cholesterol, it has deionized lipid, and it has a polyethylene glycol. All those components can induce the allergy, and that is not very peculiar for the COVID-19 vaccine. That is applicable for flu, that is applicable for other vaccines. At the moment, well, actually, that was the first thing when I told to Independent Sage that instead of passing a blanket statement that allergic reaction was induced by these vaccines, you could have tested those 2 people against different allergies, and you could come up with that, okay, these 2 people are the one that have these particular allergies, therefore you have to be cautious around that. So that has created really a problem. But in the US, they didn't really much bother about this one. So it's certainly not really a concern moving forward, because ultimately they will find out which the allergies is there. Other one was about
the one was to do with if
dosage.
you delay the vaccine or you miss it after 3 weeks.
Yeah. So the purpose of 2 doses is that when you give the first dose, our immune system sense it and prime the immune system. So some of the memory cells are there. As soon as you give the second dose, our immune system will come as a booster. So that's what we call the booster dose. Ideally, it should be given within the time it has been tested. So, for example, Pfizer BioNTech vaccine has to be given within the 21 days of the first dose. But if it is 1 or 2 days up and down, is not really a problem, because the window for example, for AstraZeneca, when they did the trial, was around 21 to 28 days, and it didn't really make big difference. But of course, the priority should be given so that you can get the right dose at the right time, so that the immune response that is supposed to be mounted, that should mount, so that you have the protection against the infection.
Fantastic. And I guess we're just being mindful of time, and we've had so many questions come through, and we will try to endeavor to answer those in due course, but it'll probably be done after the event, and we'll try to at least share it in the description on the YouTube video. I'm going to try and pose this as a last question. So I'll ask yourself, Dr. Zaidi, but all those people that are still here, please do check our YouTube link, do subscribe, do share the video. We want to try to educate and inform as many people as possible. I guess that's clearly the ethos of this event. So Dr. Zaidi, if I come to yourself, what can we learn from other countries? Let's say, for example, Australia or New Zealand or some other South Asian countries where the rate of mortality is very different to the United Kingdom and conversely, especially the United States of America. So vaccinations are a key component, but how much behavior change can we actually learn?
Well, I think behavior change is a very strong component of controlling this pandemic. And also you have to look at the population in terms of its risk factors and its demographic makeup. So like Australia, I saw that people are asking about Australia and New Zealand, where the countries are now virus free. Well, they had very strong lockdowns. They had very strict rules and regulations around wearing masks, around behavioral change that actually, this virus, it's a virus, it's an airborne virus, and for it to stop transmitting it requires very strict policies, especially given that many of us who may not even be aware that we are transmitting this virus, either because we're asymptomatic or most likely we're pre-symptomatic, and we're not wearing a mask, you're transmitting it. And super spreader events, as Dr. Munir has pointed out, are a huge reason for why this virus gets transmitted. And sadly, in the United States, the whole public health approach has been politicized beyond means. I mean, it's very sad that where you could have dealt with this virus by putting on a mask for 100 days and not having huge events, etc. We've had a government that's chosen to ignore all the public health advice. And so for that reason, we haven't been able to bring it under control. And that's why the vaccine trials have been so successful, that we have so many alternatives for vaccines, because many countries have this virus transmitting itself, It's just a confluence of, disease X was always going to emerge. There's been, for the last 20 years, we've all been waiting for this disease X to come, and it's arrived, okay? And in some ways, you could look at it, the glass half full or half empty. I'd say, sadly, we've not been able to bring this virus under control, and there have been many unfortunate deaths that should not have happened. But at the same time, the vaccine development trajectory has been absolutely phenomenal.
Yeah, no, thank you, Dr. Zaidi. And it's clear, reading from some of the comments, that people have either experienced it or lived through family members that have gone through it. And unfortunately, some have actually lost family members. So it's clearly a difficult thing to talk about for some. Dr. Munir, is there anything else that you'd like to add?
And not particularly, but I just want to pass finally a message to all our listeners who are still there, is that although we have a very good news about the vaccine efficacy, 90% vaccines, we have very limited number of vaccines that have shown that kind of efficacy. For example, polio, measles, mumps, and all those were very successful vaccine. So that is certainly a very good news. However, this is not something that we should start celebrating. There is a lag time, and the time is that until we don't reach to herd immunity, please do follow all those public health principles, those being set up for everybody, because not only that the developed world, but the challenge is the developing world. According to a calculation, and WHO has embraced that, until 2024, most of the developing countries would not have the vaccine availability. And by that time, I'm sure the disease would be there out in one corner or other. And until not everybody is safe, nobody is safe.
Yeah, totally echo those. And I would also like to add that people should definitely keep, physically distance and wear masks, because the long-term consequences, as we're learning more about it, they can be, fatigue, fuzziness in the head. People are beginning to see that. And similar to other autoimmune diseases, people often, as you yourself are a psychiatrist, don't give much credit to people when they describe these sort of vague syndromes, but people are experiencing those. So it's best to be safe and to protect yourself.
Thank you. That's a lovely way to finish and round off the evening. I'm going to stop